So,
Hello,
Mavis, please. So,
Dr. Lee.
I would like to introduce Mavis Tan. He bioinformatician from ONT, the Oxford Nanopore platform. So, and then he also have I forget the name of the guy from BCA. Yeah. Um, so he will support Mavis to answer some questions on. Yeah. So now everything is your Mavis, please share your screen.
Okay, thank you, Dr. Lee. Please let me share my. Um, Before I share my screen, may I confirm that can you hear me okay?
Yes, I'm hearing well. Yeah.
Okay, thank you so much. Please allow me to share my screen.
Yeah.
Um,
I
may
can
I
see
confirm
your
this my
screen.
screen?
Yeah.
Okay, thank you so much. So hello, everyone. Thank you, everyone for taking the time to join us. Um, So I think I will turn off my camera because in order to ensure that throughout the presentation the internet connections can remain stable. Okay, so okay, so again thank you everyone for taking the time to join us. I would like also to extend my gratitude to the organizing committee, Dr. Lloyd and his team, for inviting us to share the applications of the microbial genomics with Oxford Nanopore. So my name is Mavis. I'm the clinical sales specialist at Oxford Nanopore Technologies. And today I'm very exciting to discuss like how Oxford Nanopore is shaping the future of microbial genetics. And also joining me is my colleagues, 10, a few application scientists from BCE Vietnam, who is our channel partners in the country. And throughout the presentation, please feel free, to type any questions that you have in the chat, either in English or in Vietnamese. And my colleagues, Tan, will help to assist in answering the questions. So let's start with the overview of nanopore sequencing for microbiology and infectious disease. So Oxford's nanopore sequencing platform is transforming the microbiology and infectious disease application. Our platform provides a comprehensive and versatile solutions for uncovering the microbial world with the application range from whole genome sequencing of the microbial isolates all the way to metagenomics. And let's break down into key application. First of all, microbial isolate whole genome sequencing, where this allows a rapid and accurate pathogen identifications during outbreaks and detect the resistant genes in real time. It gives us clinicians important information to inform treatment decisions. And additionally, the whole genome sequencing with Nanopore can also helps to understand the genetic diversity and also evolutions of the microbial populations, and it can guide the infectious control and the surveillance efforts. For full-length 16S, our technology enable the sequencing of the full-length 16S genes. And with long reads, it provides a high taxonomic resolutions. It offers a greater accuracy in microbial profiling compared to short read sequencing. And the ability to sequence multiple species in a single sample also allows for the detection of the polymicrobial infections, which are often missed by the traditional methods. Targeted viral whole genome sequencing and AMR. So with Nanopore, you can also classify both known and novel viral variants, which are essential for the monitoring of the viral evolutions, as what we've seen with the case of the SARS-CoV-2. And this can be applied in detecting the AMR as well. And by integrating the sequencing into the public health strategies, our platform actually can assist in tracking the viral outbreaks, guiding the vaccine strategy, and understanding viral transmission dynamics. And lastly, metagenomics. I think this is a very powerful tool to perform a microbial profiling because it doesn't require a prior knowledge of the pathogens. And at the same time, it also allows a rapid AMR profiling. So from the metagenome assembled genomes, MAX, you can assemble genomes directly from complex microbial communities, provide insight into the unculturable or the novel organisms. So what features of our platform make it powerful for implementations of microbiology and infectious disease applications? So first of all, our instruments are highly accessible. Historically, before Oxford Nanopore technology, owning a sequencer require a significant investments. However, with Oxford Nanopore, you can start owning a sequencing capacity with just a few $1,000s. We offer the MinION, which enable a smaller labs to perform microbial genomics on site or even in field settings closer to the sample or outbreaks. And for the labs with higher throughput needs, we provide instruments like a GridION that you see in this image here, as well as a PromethION series. And our library preparation protocols, such as rapid barcoding kit, are very streamlined and require very minimal hands-on time. If you combine this with the ability to acquire sequencing data in real time without the needs for the fixed runtime, as long as you gather sufficient data to answer your questions. So this helps in deliver a rapid turnaround time from sample to answer. And lastly, as you can see here, long-read sequencing technology often face some limitations compared to long-read sequencing technology, particularly when it comes to bacterial genome assembly. So short-read often results in a fragmented assembly with multiple contigs due to their inability to resolve repetitive regions, complex structural variants, as well as the genomic region with very high GC content. And additionally, bacterial genomes also often carry plasmid, which is the extra chromosomal DNA molecules that are crucial for antibiotics resistant and virulence. And also with short reads assemblies, frequently they will miss or misplace plasmid sequences, leading to incomplete or incorrect reconstructions. On the other hand, long read can capture the entire plasmid in a single read. This will enable a clear differentiation between the chromosomal and the plasmid sequences. So this improves the plasmid identification and characterizations, which is particularly important for understanding horizontal gene transfer and antibiotic resistant mechanisms. I believe many of you may know that nanopore sequencing has been serving as a critical tool in pathogen surveillance. Even before COVID-19, nanopore has already played a key role in global pathogens monitoring. So for example, two early high-profile efforts successfully used nanopore for real-time sequencing of the Ebola virus in Africa, as well as the yellow fever virus in Brazil. And also to address COVID-19, the Africa CDC, in collaboration with Oxford Nanopore and other partners, have made significant investment to support the genomic surveillance across the continents. And this effort has led to the sequencing of over 100,000 of SARS-CoV-2 samples, with half of these 100,000 being sequenced with OMT platform. And additionally, more than 200 students and technicians were trained in the wet lab as well as the bioinformatics. And there are also many studies has now emphasized that the public health benefits of pathogen surveillance are greatly amplified when the sequencing can be conducted locally within the countries. So this has highlighted the importance of continued investment in pathogen surveillance infrastructures, such as the expanding of the local nanopore sequencing capacity. And beyond the current pandemic, right, now that we are here, the building and maintaining a robust sequencing capacity for both known and emerging pathogens will be crucial in safeguarding the global public health. And by investing in local capability, we empower the nation to respond very quickly and decisively to the future public health threats. And before we jump into the technology side of things, I would like to share a few use cases that I think underscore ways the community is implementing Nanopore for rapid outbreak response today. So on the left-hand side, you can see that looking back to mid-May 2022, many of you may remember multiple cases of mpox that started to be identified in several non-endemic countries. And on the May of 19th, Joanna and her team at the NIH Portugal have performed a rapid metagenomic sequencing with Nanopore. And this has resulted in the release of the first genome of the virus associated with this major outbreak, and then followed by the CDC using a similar approach on the Nanopore and upload the genome the next day. And in the late 2022, several European countries, including in the UK, have observed a surge in the case of the group A streptococcus. And the team at the Guy's and St. Thomas Hospital in the UK have been using the whole genome sequencing to study the bacteria isolated from the infected individual, where we will look at the case study later. Um, because by doing so, they aim to understand the epidemiology, such as the pattern, the causes, and the effects of these health conditions in the populations. However, I think the key point here is that they use on-site nanopore sequencing, which allowed them to perform genome sequencing in real time as the outbreak was still ongoing. So this is in contrast to the traditional approach where sequencing might be done later after the outbreak has subsided. So the ability to conduct on-site real-time sequencing with nanopore technology during the outbreak has allowed the team to act very quickly rather than waiting for sequencing results after the incidents. So this means that they can potentially implement a faster intervention, improve the infectious control, and also possibly to reduce the number of cases or prevent the outbreak from getting worse. And finally, we also see that Dr. Eric Carlson in Cambodia, who reported the full viral genome sequence in less than 24 hours from a human infections with H5N1 avian influenza. So he actually leveraged this quick turnaround time with nanopore sequencing infrastructure that was initially put in during the COVID-19 time. And here we also see that there are um, many recent results from the community that have demonstrated like how our lattice chemistry is delivering a very high accuracy for a range of microbiology and infectious disease applications.